Thanks everybody for joining us on the Using FCS Express Image Cytometry for Analysis of Amnis Datasets webinar. Today we're really happy to announce direct compatibility of Amnis data files in the DAF and FCS file format, or DAF-CIF format, with FCS Express Image Cytometry. Today's presentation will focus on working with Amnis-derived datasets in FCS Express, as well as quick reporting and batch analysis of those results. We're going to bring you through working with plots, opening up DAF and CIF files, working with images, and working between images and plots and the raw data in FCS Express. We also have on the line today the team from Amnis, part of Millipore Sigma. Brian Hall from that team will be presenting first on some of the unique capabilities of the Amnis instruments with some specific examples of how the workflow between IDEA software and FCS Express Image Cytometry can be used to really benefit any level of user, any scientist in any industry. So with that, I wanted to bring Brian on the line here and flip the presentation over to him. All right, Brian, the floor is yours.
Okay, great. And thank you for the introduction there, Sean. Now, my objective today in this webinar is to go through some of the basics of the Amnis technology. I understand there are a lot of FCS Express customers out there, so I'd like to introduce you to what image flow cytometry is and why we consider it a critical field in cell measurement and cytometry. So essentially, Amnis has developed an imaging flow cytometer. We have two systems, the FlowCyte, which is a 20x imaging system, and the MAR2, which is a 20, 40, and 60x imaging system. And the systems collect bright field, dark field, and up to 10 fluorescent images at a rate of around 5,000 cells per second. This image database is then taken into IDEAS, which is a statistical image analysis software package, and we use the software to quantify fluorescence intensity as well as the morphology of the spatial location of that fluorescence of each of the 10,000 or 100,000 images in the data set. One of the advantages of our approach is you have a click on the dot in a dot plot, you can see the image, and this technology enables novel applications such as measuring and quantifying colocalization, apoptosis, looking at signal transduction cascades, as well as morphological cell classification. And it's really agnostic to discipline, though it's broadly applicable to many research fields. So the way that you can think of the Amnis technology is it's a flow cytometer is very fast, it's photonically sensitive, and it's quantitative, but it lacks the spatial resolution that you get with imaging. For those types of experiments, you would typically use a high-resolution confocal microscope, but the problem with typical microscopes is they lack throughput. You typically are looking at 1 to 10 images, potentially 100 images, so it's not as quantitative as flow cytometry. So the Amnis systems are really in a unique niche between these two technologies where we have the high throughput of flow cytometry combined with the quantitative and morphological aspects of a microscope. Now, the platform is designed to image all of these images simultaneously. What that does is enable our throughput, again, up to 5,000, potentially 10,000 objects per second. What you're looking at here is just a cartoon of the optical bed. So samples are prepared in suspension, just like you would for flow cytometry. The cells are then taken into the system. They're hydrodynamically focused in a quartz cuvette. And we use a series of excitation lasers to excite fluorescence and a series of LED colored lights to illuminate bright field from behind. The image is captured with either a 20, a 40, or 60x imaging objective, and that image is projected through a series of Petzval lens sets to what we call the spectral decomposition element, or the filter stack. And the filter stack spectrally decomposes that image into its constituent wavelengths. So if you look at this magnification here, we get the blue, green, yellow, orange, red, and far red images lined up next to each other. And all of these images maintain spatial registry. In other words, if we overlay these three, all of these images, they maintain spatial registry so that we can measure the distances between these fluorochromes. By looking at each channel individually, however, we can quantify the total fluorescence. So this gives you a basic overview of the optical layout of the system. Now, the camera that we're using is a CCD camera. We're going to operate that camera in what's called time delay integration mode. And what time delay integration enables is for us to capture images with high resolution up to 0.3 microns per pixel and high photonic sensitivities, photonic sensitivity that rivals a flow cytometer. And the way that we accomplish that goal is rather than take a static image of the cell as it's streaming through the instrument, if you look at this animation here, as the cell is transiting the detection region, we actually integrate the light from the pixel above to the pixel below in exact synchrony with the transit of the cell, and then read out the last pixel row and reconstitute the image of the cell in the computer database. And what that allows us to do is increase the integration time of the fluorescence so we get high photonic sensitivity. But because we're transiting that energy from the pixel above to the pixel below with the velocity of the cell, we maintain a 0.3 micron per pixel resolution. So it's a way for us to, again, get high photonic sensitivity with high image quality. And this allows us, with the spectral decomposition element, to capture 12 images per cell at a rate of 5 to 10,000 objects per second. So it's a very high throughput. Now, the raw data looks something like this, and what you're looking at is in a single row here, you have an individual cell. We have two bright field images that enable us to do cross-camera registry. And this particular cell is labeled with FTSE, PE, DAFI, and Alexaflor 647, and you can see there's a number of blank channels in here where I could have used other fluorochromes, so you can do up to 10 colors of fluorescence. Now, the software allows you to create any combination of overlays, so you can look at the individual fluorochromes lined up next to the bright field image, and then on the far right here you can see a composite image that shows an overlay of all the fluorochromes simultaneously. So you can look at spatially where all these fluorochromes are spatially located. Now, the big question we get asked is, why is imagery important in my flow cytometry experiments? And what is the real benefit that imagery brings to flow cytometry? And I like to use this example where by flow cytometry you essentially have something like this, which is a dot plot. And in this case, we're looking at DAFI fluorescence versus Alexaflor 647 fluorescence. Now, in ideas, when you click on a dot, it allows you to see the image of the cell located at that location. So by clicking through three individual images, you can see that all of these cells have exactly the same total fluorescence intensity. So by flow cytometry, all three of these cells would be located in a single population. So we would draw a region and say all of these guys have 244,000 counts of Alexaflor 647 intensity. But if you look at the imagery, it's pretty clear that these cells are morphologically distinct. So by using computer algorithms to quantify these morphological characteristics, we can see that these three cells morphologically are in three very distinct populations, where the top cell is located on the left in the small population, the middle cell is there in the right to the in the larger population here, and the cell on the bottom, the conjugate cell, is down in here. So using computer algorithms and quantifying that morphology, we can add benefit to our flow experiments. Now, IDEAS also allows you to calculate different types of intensity. So here we can use max intensity or the mean intensity, and by looking at these features, we can actually quantify where this fluorescence is spatially located. Now, another advantage of our approach is that we can leverage what's called masking. And all a mask is it's a way to identify a region of interest within the cell. So if you look here in this top example, you can see the top image has two nuclei. These blue pixels in between are masking the region between the two nuclei. And if we take this mask and apply it to my actin fluorescence, you can see we can quantify then the total amount of fluorescence located at the juncture between these two conjugates. Now, in the top image, there's a fairly low amount of actin fluorescence there. But if you look at the bottom image, you can see this image is a T cell binding to an antigen-presenting cell, and that actin has actin has been recruited to the immune synapse to support communication between these two cells. And so if we take this mask and overlay it on the immune synapse here, we can quantify a much higher total fluorescence intensity of actin at the immune synapse as opposed to the rest of the membrane of the cell. And this really gets to the heart of why adding imagery to flow cytometry data is important, because it allows you to look at experiments in novel ways. So now if we take this approach to a data set where we've induced immune synapse formation within these cells, we have T cells binding to antigen-presenting cells, we now have a metric by which we can quantify the rate of immune synapse formation. And if you look at the mean pixel value of the valley mask, which is that region of the immune synapse, you can see that about 70% of these cells have proper immune synapses, and that the remaining 30% of the cells have uniformly distributed actin, or potentially these are just coincident conjugates. So it's a way to quantify this type of morphological characterization. Now, the image stream can be used for a number of different types of applications. Some of our core applications include cell signaling, things looking at NF-kappa B or FOXP3 localization as cytoplasmic or nuclear, identifying internalization and phagocytosis either in exosomes or in yeast or bacterial particles, looking at co-localization, how molecules are internalized and trafficked within the cell either through an endosome or a lysosome. We have groups that are looking at overall shape changes, not only in looking at immune response with MCP1 activation of monocytes, but also looking at unique red blood cell morphologies and other types of cell morphologies that are important for different experiments. I talked a little bit about the immune synapse formation, but characterizing cell-to-cell interaction, looking at apoptosis and cell death or autophagy, cell cycle and mitosis. There's many groups in the field of stem cell biology that are using the image stream to characterize differentiation of various stem cells. And there's other groups as well, microbiology, parasitology, and there's a big oceanography group that use it for looking at marine samples. So the image stream is broadly applicable to a number of different research fields. And really, the strength of the system is when you're trying to quantify total fluorescence intensity and look at unique morphologies at the same time. So a good example to highlight sort of the benefit that imagery brings to flow cytometry applications is this one, which is looking at quantifying the apoptotic index. And in this case, we're looking at Ramo cells that were treated with 5 micromolar camptothecin to induce apoptosis. We then labeled the cells with FIFI-LUX, which is going to identify active caspase 3 as an indicator of apoptosis. Annexin 5, which is looking at depolarization of the phosphatidylserine residues, and also DRAC5, which is able to label both live and apoptotic nuclei. So we chose DRAC5 so that we could get morphology on the live cells. In this experiment, we're going to collect 20,000 events and quantify the apoptotic index using active caspase, annexin 5, and nuclear fragmentation. And what this allows us to do is look at the orchestration of the apoptotic pathway within the cell type. So the first plot that we would look at is looking at the active caspase 3, and you can see the untreated sample has about 30% active caspase. You look at the associated nuclear morphology, you can see we have nice round nuclei here. After 6 hours of camptothecin, we get about 70% of the cells are apoptotic. We have fragmented nuclei. The cells are simultaneously positive for annexin 5, as you would expect, and they're bright for caspase 3, indicating that these cells are apoptotic. If we do the same flow cytometry type analysis of looking at the annexin 5 dot plots, you can see the untreated is about 4%, untreated, and that goes to 77% as the apoptotic index after 6 hours with camptothecin. And then what we can do is look at these two measurements, the Nexin-5 and the caspase-3 simultaneously, to characterize which of these markers is identifying which cell type. And if you look in the lower right, now these are cells that are positive for an Nexin, or for Phi-Phi-Lux, but negative for an Nexin-5. In the upper left, these cells are positive for an Nexin-5 and negative for active caspase. And one way to explain this sort of difference is that it's possible that caspase-3 is an early indicator of apoptosis, and that this 4% here are the early apoptotic cells, this 10% here are the late apoptotic cells, and then the vast majority of these at 6 hours, about 70%, are sort of mid-phase apoptotic. What's interesting, though, is if you look at the double negative population, you can see there's a population of cells that are negative for both of my apoptotic markers, but still have fragmented nuclei, indicating that these cells are in fact apoptotic. But in the absence of nuclear morphology or the imagery, we would have no idea that this apoptotic population existed. So what we would do next then is use a computer algorithm to go in and actually quantify the morphology of the nuclei as a measurement for apoptosis. And we can do that by looking at the IDEAS features contrast versus the area of the condensed chromatin structure. The live population, they have nuclei that look like this. They're nice and round. And then after treatment with camptothecin, you can see we get nuclear fragmentation and blebbing in the bright field imagery. And so we could use this morphology to characterize the apoptotic index. And when we do that, we get about 90% of the cells are apoptotic by morphology. Now, the way that our system works is we use what's called a feature, and a feature is used to quantify a unique morphology. The two features that I'm using in this experiment to measure apoptosis are contrast, and the way contrast works is it measures the texture of the image. If you look at this far left image here, what I've done is I've used a line to transect and produce a histogram of the pixel values under that line. And if you look at a normal live cell, there's very little blebbing and very little contrast within this image. When you compare that to a highly apoptotic cell that has lots of very dark regions and lots of very bright regions, as the cell increases its texture when it enters the apoptotic pathway. And so contrast in the bright field image is an effective way of looking at apoptosis. Now, the gold standard for measuring apoptosis is nuclear condensation and fragmentation. And to do that in IDEAS, we can calculate a mask that measures the brightest 50th percentile of the pixels in the image. And those are these blue pixels here in the image. And then calculate the area of that so that as the chromatin structure condenses and the nucleus starts to fragment, which is a hallmark of apoptosis, we can quantify that event. And so if you have small areas and small nuclear areas, then you're likely an apoptotic cell. And so this just gives you an idea as to how some of the morphology-based features work in IDEAS. And then we can use those features to quantify a time course. And here you're looking at apoptosis as measured with camptothecin and Nexin-5 and nuclear fragmentation over a 24-hour time period with 6 different time points. And you can see nuclear fragmentation in red here is actually a very good indicator of the apoptotic index. So that just gives you a general idea as to how a computer algorithm can be used to measure unique morphologies. But IDEAS essentially has 86 features per channel with 22 function masks and the ability to generate user-defined features. And so there's essentially a huge feature data set that you can use to quantify any type of morphology within your experiment. So with so many options, how do you verify that the approach that you've taken is the most appropriate? You can look at up to 2,000 features and IDEAS. You can create new masks and identify different types of size, shape, and signal strength features. You can combine features. So there's a powerful ability to discriminate different cell types based on their morphology, but determining the best feature can be very difficult. And so to assist first-time users in doing this type of technical quantitative measurements, Ideas implements a wizard workflow where we have wizards for some of our core applications, such as apoptosis, cell cycle, co-localization, internalization, and nuclear localization, as well as spot counting and shape change. But for applications that are not in the core feature set, we use what we call the Feature Finder wizard, which is a wizard that you can implement to hand tag two unique morphologies and then have the software tell you which is the optimal feature in ideas to analyze that particular experiment. So again, this is really agnostic to any of these core applications. It's open for you to apply it to any type of experiment where you think you see a morphological difference between either the fluorescent staining pattern or the morphology of the cells. And so the Feature Finder is sort of a catch-all wizard that allows, enables many different and diverse applications. So recently we've been teamed up with the group at Novo Software, and what they've been able to do is implement a way of analyzing the image stream data so that after you're done with your ideas analysis, you can use the software to create a high quality graphic presentation of your data. The software enables you to load imagery as well as look at many different types of dot plots. And what's really great is you can also look at DNA, cell cycle modeling, proliferation modeling, and allow you to batch process your presentation so that you can rapidly export multiple dot plots and histograms, and it gives you live updating of graphs so that as you change your regions, all of the data files update seamlessly. So it's been a great advantage to work with the group at Novo Software and FCS Express. And I think with that, what I'll do is turn it over to Sean, who will give you a detailed demonstration of how the FCS Express with the image analysis works on ideas data sets. And so, Sean?
Hi, Brian. Thanks for that great presentation. We just want to take a moment here and pause to see if there's any questions from the audience about anything related to the Amnis and the imagery and image cytometry side of things. So if you have any questions, just enter them in the question and answers box. We'll try to answer them on the fly here or come back to them at the end. So again, we're just going to kind of switch gears here and move to talking about what you can do with that data that you generate from ideas in third-party tools like FCS Express. FCS Express and the team at Novo Software, again, we're really happy and really excited to be announcing direct compatibility with these ideas-derived data files, not just exported FCS files. We're talking about the directly derived DAF and CIF files and the imagery involved with that. FCS Express is the only third-party software on the market now reading those data files. So what we're going to do in this portion of the webinar is show you how to load those data files up in FCS Express, how to create a basic analysis from those data files, how to create some plots, how to work between the raw images from the ideas software, the raw data points, and all the kind of advanced plots and features that we have within FCS Express. We're going to do this with a cell cycle data set, so we can show you about a little bit more about the advanced functionality for things like cell cycle modeling. We're also going to give you a little example that has something to do with the apoptosis data sets that Brian was showing in the previous portion of the presentation. If you remember, he kind of showed you a few kind of nice regression curves that were generated from a multiple time point series. And what we're going to do is show you in FCS Express how you can actually calculate those regression analysis directly within our software as well. So you have access to the raw images, high quality, super resolution plots, as well as live updating regression analysis for the data sets. And some of the key things to remember about FCS Express, if you're not currently an FCS Express user, FCS Express has this Microsoft Office look and feel, which makes it really easy to use and learn. And in fact, we're within FCS Express right now. So this presentation, this webinar is being given within FCS Express software. For those of you coming from ideas and you need the ability to create some additional visualizations, some kind of high quality reports, and kind of seamless exports to PowerPoint, to Excel, to PDF. We can do all that directly in FCS Express, which is a kind of nice thing and falls in line with the collaboration that we have with Amnis. FCS Express also allows you to work not just with single files. We can work in plate-based mode for high content data, where you could simply load in hundreds or thousands of individual data points if you want. We can batch process and quickly analyze that. And for those of you that are coming from a more regulated environment and you need 21 CFR Part 11 compliance features, well, FCS Express and Novo software can help you facilitate that. We have some add-ons available to the software for security and logging and audit trails that will help you get to the level of compliance that you need for your regulatory or regulated environment. Now again, some of the unique features for FCS Express that ideas might not offer the best solutions for, but FCS Express does, and we've developed them for many, many years, are batch processing, histogram overlays, working with various modeling such as cell cycle, proliferation analysis, kinetics, this integrated regression analysis I talked about, and a lot more in there, of course. So without further ado, I'll show you how we get some of these data files loaded from Amnis and ideas into FCS Express. We're going to do this by starting with what we call a layout in FCS Express, which is essentially this blank page. Again, this is going to look a lot like a PowerPoint presentation, and it's also going to work a lot like a PowerPoint presentation. So if you're new to FCS Express, don't be nervous that it will be difficult to use. When you create a PowerPoint presentation, you will do things like inserting shapes, text boxes, and pictures. We can do all that in FCS Express, but then we're going to come over and insert objects like histograms, multi-cycle DNA plots, dot plots, density plots, contour plots, all the things that you need for advanced flow and image cytometry analysis. So to get started with our Amnis data, I'm going to choose to insert a color dot plot to start. And the first thing that FCS Express is going to do is it's going to prompt me for what data files I actually want to load up. So on my other screen, I'm going to bring this over so everybody can see it. We are being prompted to select a data file, and you can see here that we have two DAF files in this particular cell cycle example folder. And in this version of FCS Express that we're running, you can see that we have Amnis DAF file integration. So this is now a new file of type. This will be available in the next major release of FCS Express, which is coming up sooner than later. We can give you some more information on that after the webinar. But again, you need to choose Amnis DAF files, and you can pick your Amnis DAF file, click Open, and now we've inserted a color dot plot for that particular data set. And just like any other plot in FCS Express, you can move this object around, you can resize it. Really anything that you can do in PowerPoint, you can do with this particular plot or object. Now the other big thing you're going to be wanting to do with your Amnis data is looking at the images. We handle working with the images in FCS Express through what we call a data grid. This may have some changes coming forward to what we call this, but this is the functionality you'll need. We insert a data grid, and you can see that in this data grid, we've displayed all the different cells that are associated with this acquisition. And you can see here there's about 3,000 cells. We've displayed all the different parameters for this acquisition, and all the way at the end of this list, you'll find that the images appear. And these are all the images that were acquired at the time of acquisition with the Amnis ideas software and image cytometry. Now there's some ways that we can make working with this data grid a little bit easier. We're going to go into a few of these different features as we begin to build an analysis. But really, if you want to format this data grid, you can right-click on it and choose Format. And in our parameter to display options, we have some nice options here at the bottom for just picking and choosing what you want to show. So instead of showing all of the parameters that you've acquired. Maybe you just want to show a few of the images. Well, we can do that. And now you have a nice data grid here just showing you the images associated with this experiment and which cell they're associated with. Now at the most basic level in FCS Express, when you need to move between your data grid and any of your plots, you can simply create a gate on your analysis, and we'll show you a little bit more about this in just a moment. But when you have this gate created and it's applied to any particular data grid, and you can have a number of data grids on your layout, this will all update in real time. You can see that as I adjust this gate, we're showing you all the events and all the cells and all the images that fall within that particular gate. So there's a lot of unique things that you can do with this as you're working with your analysis. You get that access to the morphology that Brian mentioned is really important for working with these data sets. With FCS Express now you get the access to the raw data. You can seamlessly work between the two, and we can show you now how to kind of take advantage of some of the high resolution plots and batch analysis features within FCS Express. So let's go back a few steps here. I'm going to show you how to create an analysis in the software. To go back a few steps, it's very easy in FCS Express. We have convenient undo and redo buttons up here in the upper left. You can see as I undo, we're moving that gate back around, or if I redo, removing it. Really where I want to get us back to, and kind of show you the power of this undo button, is where we started with this initial dot plot. Now the other plot type I want to insert here is a histogram to help me start my analysis. So let's insert that histogram. Let's move our objects around so we can really start doing some serious analysis for this webinar today. Now, when you have a plot inserted, you can pick and choose which parameters you want to view on that plot very easily. You can click and hold on the axes, and you can either scroll through the axes, or if you begin typing the name of a parameter, in this case I know I want to use the gradient RMS parameter, it will jump you to there, and you can move to that parameter of interest. So in my second plot here, I want to move to an aspect ratio for MO1, and then look at our area for MO1. Now generally when you're beginning analysis in IDEA software, you do some basic gating to eliminate kind of debris, large cells, maybe over imaged or overexposed type of things. So we can do that in FCS Express, of course. To create a gate, you simply click on the gating tab, you choose the gate type that you'd like to draw, and then you can draw that on your plot of interest. When you have that gate drawn, you'll be prompted with the gate view here with a create a new gate dialog, and you can give it a name, and I'll call this region 2. You'll see why I skipped region 1 in just a moment. But as you can see here, we now have the region 2 gate created. Any of the cells that fall within this gate, because it's a color dot plot, will appear as a color. That means we have live updating, backgating within FCS Express, as well as live updating statistics. So as this gate is adjusted, you can see the statistics are updating. Or even if we apply a gate to another plot, again, you'll see as we adjust that gate, everything is updating for us in real time. Now the other type of gate that you may want to create, and you're probably used to doing in IDEA software, is creating a marker on a histogram. This is useful when you're looking at this first gradient RMS parameter here. We will create a marker on this particular histogram, and what we'll do to that marker is now convert this into a gate. So we'll right-click on this plot, convert this marker and link it, and that will prompt us again with another create a gate window, and here we'll call it region 1. So you can see here now that I have events appearing on my dot plot on the right as blue, and as I adjust this marker, you can see that the backgating is updating in real time. So another kind of really useful feature for working with FCS Express. Now I'm also going to come to the Insert tab and insert what we call a gate view. This is going to help us keep track of any gates that we've created, as well as keep track of the hierarchy and change the hierarchy of our gates at any time. So if I want to make region 2 a child of region 1, I simply drag it onto the region 1 gate. You can see that the hierarchy has updated. It's updated our plots and our backgating here. We'll do an undo and redo that real quickly so you can pay attention to how that backgating is changing, and you can see now that it's quickly been updated. Now, if you'd like to apply a gate to any plot, you saw kind of earlier I dragged and dropped a gate from one plot to another to apply it. Another way that you can do that is simply dragging from the gate view here onto a plot. You can see now that the region 1 gate associated with the marker is applied to that. And in the gating tab, you can even choose here from the gate dropdown or remove the gate. One of the features I really like in FCS Express is that if I know I want to open up this region 2 gated population into a new plot, well, you don't just have to insert a plot and apply the gate. It's a lot more straightforward. You can simply drag this gate out, drop it onto the layout, and a new plot will be opened that has that gate applied to it. So when I'm ready to change parameters here to say my intensity for MC2 versus my intensity for MC11, you can see now that the region 2 gate is applied. As I update that, it will be updated in real time. Now, again, when you're working with some of the Amnis data, if you're not used to working with the data scaling that's in there, maybe you're used to working with flow cytometry data and you're just kind of coming over to the light in the image cytometry world, well, the scalings can differ a little bit. In this case, FCS Express has a great feature. If you right-click on a plot and choose Magnify Resolution, you can simply zoom in on a population of interest or an area of interest on that plot. It will zoom in. You'll have access to that data a little bit more easily. Now, another feature that I know a lot of people who are using ideas would like it a little bit easier is being able to change plot types, kind of changing between contour and density plots. You can see here in our Insert tab, we have a lot of options here. But if we come into Format in FCS Express, we can change to any of the plot types of interest. So if we wanted to move to a contour or to a density, really whatever is going to help us visualize this data most appropriately, we can change to that plot type. And in this case, I'll just use a contour, which might be a little bit more appropriate. So the last thing I want to do here in our analysis, or the last gate I want to create before we kind of move on to the next phase of working with the cell cycle data, is this region 3 gate. Again, if you're used to working with Amnis data, this might be a kind of familiar gating strategy that you've set up. And at this point, we're ready to begin looking at the cell cycle parameter. Again, this is a cell cycle analysis data set. The cell cycle parameter is the intensity MC11. I believe that's DRAC 5 for this data set. So what we'll do is insert another histogram and change to the intensity for MC11. And you can see here that we have a DNA cell cycle styled histogram. Again, our scaling, it's kind of scaled down here on the low end. So what we'll do is just simply magnify this resolution to make it fit our plot area a little bit better. And again, we probably do want to apply a gate to this. You can see what happens here if I apply region 3 from our gating hierarchy. It cleans up the data set a little bit, takes out these events that were falling out here. And again, one of the nice things we can do here is if we need to find out what was going on with those cells, well, we can simply insert this data grid again. Let's choose to view a few of the images here for DRAC 5, NF kappa beta in the bright field. I'm also going to do something here where I'm going to show gates. So it's going to give us some backgating information for those cells. And we can find out now if we wanted to, again, kind of quickly create here a marker on this particular region, convert that marker to a gate. We will put this over here as gate 1. And we can quickly review here and see what are these cells that are actually falling up here. So you can see some of these, they might be dividing, they might be outliers. We probably do want to exclude them. There's not too many. And if we get back here and we take a look at kind of what's going on with the rest of our analysis, we can see that these are probably events that we want to analyze. Again, you can always go back and find out what about the morphology is most interesting for you for that as well. So for now, we'll just remove that gate. And what I want to show you is one of the more advanced functionalities of FCS Express for working with the cell cycle data. As Brian mentioned in the beginning of his talk, what we can do in FCS Express is insert what we call a multi-cycle DNA plot, or we can convert any existing histograms that you have or any plots to a multi-cycle DNA analysis plot. So rather than trying to set up markers here and kind of eyeball where G1, S, and G2 are, I can simply come into my Format tab, choose to change this plot to a multi-cycle DNA plot, and you can see that MultiCycle has tried to fit this data, and we'll just do a quick unmagnify and zoom in here a little bit, again, to get this to model correctly. And you can see that the multi-cycle model has found the G1, the S, and the G2 phase for this particular data set. So without having to create markers by eye, we've done this more objectively, had an algorithm model this. And when you're ready to report statistics for this multi-cycle DNA analysis plot, or really any plot in FCS Express, you can simply right-click and choose statistics. So here we have some specific DNA cell cycle statistics. So if we wanted to quickly generate the percent G1, G2, and S phase, you can see that that's inserted there. Of course, you can right-click on any of these plots and get gate statistics and things like that. But there's another feature in FCS Express which is quite unique. You can actually insert a text box just like you would in PowerPoint, and you could create a report. And we can call this our DNA analysis report. And within this text box, you can place anything that you want. You can even derive information from data files. So if I drag this whole plot in there and I say, I want to put in the file name, I can do that. And now the file name will be automatically inserted, and as new data files come in, that data file name will be updated without you having to enter that information and worry about some sort of clerical error. The other thing we can do in this report is that we may want to insert a little table here. So we can insert a table. We can put some information in here, like maybe the percentage of G1, maybe percentage of G2. Drag this in here. Maybe we even want the percentage of region 3. Maybe we want some information about region 2, so we drag the whole gate in. And when we do that, you can see we're prompted for which statistic we want from that gate, so we'll just choose the number of events. And we've quickly generated a report all by drag and drop, all quick and easy to do. Now, another nice thing you may want to add to your report is representative images of your cells of interest. So if I have a few cells in here and I know that they're a G2 cell, maybe I went back and I looked at the backgating for these, what you can do is simply drag this cell out from the data grid, choose to insert a picture for it, and you can see that you now have a picture for that cell, a representative image, that you can then insert anywhere you want. Maybe you want to put your G2 cell, maybe not quite that large, but you want to stick it onto your DNA analysis plot. You want the representative image from the DRAC5 channel on there as well. We can certainly do that. So FCS Express really gives you full flexibility for what you want to do in your report, how you want to present it. We can pull from all the images that you have and create any sort of kind of customized table that you'd like for this. So once you have created a report, again, we have some basic statistics here. We have some representative images. We have our data grid that we want to put down here. We just have to find a place to put everything, just kind of like you might fight with a PowerPoint presentation sometimes to find enough room for everything. We'll just find a little bit of space down here, maybe even move our table up here, get rid of this guy. We'll find room for everything. Once we have this report populated, we can easily then export to Excel, to PowerPoint, and to PDF. So in this case, I'm going to show you just doing a quick one-off export where we say export as a PowerPoint presentation. And we will just give this a name. I will put this on my desktop. And this is on my other screen here, and we'll choose Open the Presentation. And what will happen is PowerPoint is going to open up. You've created a high-resolution report presentation from this particular analysis. We have our cell cycle modeling. We have our images. Again, if you're ready to go to a poster or a presentation, you can blow these up as large as you want. You can change the DPI and the resolution settings within FCS Express, so you can really get the resolution that you need that's in there. Now, to kind of go along with this is again a kind of single report. You never really collect data with just one sample. And in fact, even with this particular cell cycle analysis data set, you can see that I have two samples that came with this. At any time, you can pick and choose. You can click between samples. If you have multiple samples, we can run a batch process. I'm going to show you a little bit about running that batch process, as well as how to overlay some of these samples to create nice histogram overlays. So to do so, I'm just going to open up an example here. Let me pull this up from the other side of my screen here. And really what I want to show you is once you've completed this analysis and you're ready to run a batch, well, you can save this as a template, which is another kind of great feature in FCS Express. And we can simply load data files into this template. You can see here that I have 6 DAF files. They get populated into the template. And I'm just going to choose which images I want to display here, just because I might not want to show everything all the time. So I'll choose, for this apoptosis experiment, cast space, bright field, and an X and 5. And now our analysis report is done. When we want to move between these different data files, we simply click on the file of interest. You can see that it's all updating for us. And I do want to draw your attention to this histogram where we've created an overlay. We have an overlay of the first file here in gray, and now we're showing the 5th file in our data list here in red. Well, these overlays are very easy to create in FCS Express. You simply drag from the data list onto the histogram, say add the file to a plot as an overlay, and you can see that that overlay will be created. You're not just limited to 1 or 2 or 3. If you have 100 data files in here and you can compute that data in your head, you can add them all to your histogram as overlays. Now, again, when you have a lot of overlays, you probably want to look at some of the different options for visualizing these. You may want to smooth some of these, you may want to fill them, you may want to change the transparency, so that way you can make a little bit more sense of what's going on. If you need to view these in 3 dimensions, of course you can come in and click on the 3-dimensional viewer to kind of split them up. Or one of the ways I really like to work with histograms is in what we call a stacked mode in FCS Express, where we actually take all the histogram overlays, we put them on top of each other, so it makes it a little bit easier to visualize, especially when you're dealing with quite a lot of overlays that are trying to be visualized at the same time. And any of the markers that you create on these histograms will apply the statistics for every overlay that exists. So again, let's just go back a few steps here. Get back to where we started in this particular layout. We'll go back to our first iteration. And to kind of swing back around in this, what I was getting at is the batch processing functionality. So here we have 6 samples. And in our batch analysis tab, you can see that we have many actions available that you can do on all the different files that are in your data list. We have some more advanced webinars about how to set up batch actions, and I wouldn't even say advanced, they're just a webinar to explain how to do it. Essentially, you create an action for everything you want to do, and FCS Express is going to perform that for every file that's in the data list. So if you wanted to add something like the percentage of our 6 cells here to your Excel column mode, you just drag it and drop it. It's going to be added to your Excel report. It's really that easy. What will happen is when we click Run on this batch process, you'll see that FCS Express is completing X of 3 actions: export to PowerPoint, to PDF, and Excel. It's doing that for all the different iterations in the data list. So you can imagine if you had 10 files or 1,000 files, FCS Express would handle that. And it's generated a collated report here. Here's Excel. Here's the new statistic that we dragged and dropped to export into there. Here's all of our different time points and our file names. What I have coming up here is my PowerPoint presentation with all the images. And again, on my desktop here, we have a high-resolution PDF that was exported for this. So again, you can see the kind of value in being able to batch export all of these results with a click of a button. You can keep these analysis as templates, so once you do an experiment and you run it over and over again, you can reuse this template in FCS Express. Now, the last thing we want to show you today is, you're not always running an analysis with one data file at a time. And of course, with this 6-point or 6 time point apoptosis experiment, you may want to do some things like when you have that Excel report, after the fact, you may be taking that data into Excel, keeping it there, or bringing it out to Prism, or doing something, creating a regression analysis based on the time points, the treatments, the percentage of gated cells, whatever statistic it is that you may be deriving from FCS Express. One nice option that we have in the software is that you can actually create these regression analysis directly within FCS Express, and they're going to update in real time as you do your analysis. So I'm going to open one of these up here for you as an example. So instead of loading one data file at a time into a report, in this last report, or this last example we're going to show you, we've actually loaded up all 6 data files into one analysis report. FCS Express is going to load all of these up. You can see that each row in our first page of a report is a time point. So 1, 2, 3, 4, 5, 6. We've loaded up all the data files here in the data list. And conveniently on our second page, we're calculating a regression analysis, any sort of regression statistics that you want from these data sets. And as we update these gates, you can see that the regressions and the statistics are going to update in real time. So if you need to hone in on a population to see what maybe an IC-EC50 looks like for that population, maybe you need to remove a data point because you have some extreme outliers, well, you can set the gates and you can see how that updates and changes your data in real time. And there's a lot of really powerful things we can do with regression analysis in FCS Express. There's some more advanced features in there if you need to do t-tests and other statistical tests between data sets. So keep these in mind. Again, efficiency for working with large data sets and batch processing and the ability to kind of cut out that middleman or that end product of having to go out to Excel or Prism to analyze this data. And again, as we can mention, the last thing you may want to do here is go back and review some of the images associated with any of these plots. Well, you can simply do that by clicking on the plot, inserting a data grid, and you'll have access to what these images and the imagery from Ideas software had looked like at the time. So with that, I just wanted to summarize a little bit what we talked about in FCS Express today. Again, we're very happy to be working with the team at Amnis to offer this direct compatibility with DAF and CIF files. This is really a big thing for anybody who is used to using software outside of Ideas or wanted something outside of Ideas for doing some more advanced things like batch processing, easy to use histogram overlays, cell cycle modeling, this integrated regression analysis, and a lot of the other things that we offer in FCS Express, which are really quite unique to us. And FCS Express, again, is going to be able to work directly between the raw data, any of the plots, the high-resolution visualizations and displays, and the images directly. So you get that full functionality of what you're used to in the Ideas software, at least on the data analysis side of things, within FCS Express. So you can see here we have a note here that you can get a free 30-day trial of FCS Express at denovosoftware.com. I should mention that this compatibility is not quite available to the general public yet. It's going to be available in just a few months' time, most likely. So keep an eye out on the Novo software website or check your inbox for some messages from the team at Novo Software about when this will be available, when you can download it in a demo and try it for free before considering a purchase, or if you already have FCS Express Image Cytometry, how you can upgrade and get to this version that's going to allow you this direct compatibility with these files. And if you do have any questions after the webinar for the team at Novo Software, please email us at support@denovosoftware.com. So at this point, I'd like to open up the floor for questions, and thanks again for everybody who came out and joined us in the webinar today. We have Brian on the line here as well. I don't know, Brian, if you wanted to say a quick ending comment.
Yeah, no, that was great. I want to thank everyone for attending. And again, if you have any questions, the contact information is for Amnis. You can contact us through Amnis Support or email us at the Amnis Support at emdmillicor.com. And we'd be happy to answer any questions you have about the image stream.
Great, and thanks again to Brian and the entire team at Amnis for making this webinar possible today with us and also making the compatibility possible. We think this is going to be great for everybody involved on the scientific side and on both sides of our companies as well. All right, so again, we'll have the floor open for questions here for another 10 or 15 minutes.